Hello, this is Scott Carpenter, and tonight I want to start a series called The DNA Study in a Nutshell, and it's based off a presentation I did a few years back, PowerPoint, and I'm just getting deluged now with emails, questions, messages, text, and since Dr. Ketchum is, doing interviews again, the study has been rebooted with the influx of funds to be able to complete the study and do analysis and the dogman DNA, etc. Interest has peaked again, and so so have the detractors, by the way. But that's to be expected. If you're not over the target, then you're not going to get any flack. So, what I'd like to do is to go through this study. It's probably going to be a three episodes, and I'll just kind of try to work my way along. Now, this is a basic overview of the study, but it gives you good detail. It's a good explanation without getting into the weeds as far as all the scientific stuff. Because a lot of people eyes glaze over, and so, and they don't really want to hear that, but they have questions, but they just want direct, what did you do? What do you mean hair samples? What do you mean it was non-contaminated? What do you mean about this, that, or the other thing? So, the DNA study in a nutshell is the presentation I came up with, and it kind of goes through my involvement in the DNA study and also the history of the study from my perspective. And with research, information here comes from Dr. Ketchum, David Pilates, and other people that I worked with on. I was on the submitter team. In other words, I worked to gather hair samples and send them in. And then my relationship with Dave Pilates in North American Bigfoot Search, and I had that relationship with him, so I knew a few things that were going on behind the scenes, and strategies and stuff like that, at some level. Now, I wasn't involved at all on the scientific side or the scientific team. I was, like I said, I was a submitter and I was a field researcher, gathering information there. But of course, I was kept apprised and kind of knew what was going on. And I'm also friends with Dr. Ketchum. And we've talked since, we've talked off and on. I haven't talked to her in a while, but, we know each other, and we've talked multiple times on the phone. So, with all that information and the study being a public, it's on the Sasquatch genome site, and so it's downloadable, and you can go look at it, and you can do your own research. But I put together this little presentation. So I'm just kind of going to go through the presentation. Like I say, tonight will be the introduction of me blabbing on for a little while, and then part one. And like I said, it may be, I'm thinking 3 parts. We'll just see how far we get. I want to break it down. Into digestible pieces. I don't want 45 minutes, 2 hours, whatever. I want to try to keep it about 30 minutes. So I'm watching my little counter here, and I'll try to keep it right at 30. So we're going to start. Of course, back in the day, the study, was known by different names. It's the Sasquatch Genome Project. It's the Sasquatch DNA Study. Some people tried to, pigeonhole it and say it was the Ketchum DNA Study, but she didn't work on it alone. It was a team effort of multiple scientists with multiple disciplines. And the official name is the Novel North American Hominids, Next Generation Sequencing of 3 Home Genomes and Associated Studies. And that's how the paper was actually published and presented for peer review. So how it all began is in 2009-10, the TV show Destination Truth, starring Josh Gates, brought a hair sample back from Bhutan after finding a footprint. And I remember watching this episode. He found this footprint. One of the few times that they ever got anything, but he actually found a real footprint that was recently made, and not too far away from where this footprint was. They found the footprint. They also found some hair, and so this is actually from the from the TV show, and that's them at night finding the hair and collecting it. And the producers of the show approached Dr. Ketchum from DNA. She had her own lab called DNA Diagnostics, and so they approached her to perform DNA analysis. Now, Dr. Ketchum, and if you listen to any of the interviews, she really didn't, at that time didn't believe the Sasquatch existed, and had tested samples when people brought them to her, because she's an independent lab, and if you're willing to pay her, she's going to run your samples. And they'd all come up, with it'd been dog hair or horse hair, but she went ahead and ran these samples, and to her surprise, and this is a quote from her, the hair visually is not human. It's coarser than horse tail hair. And then when she compared the sequence to Jim Bank, she couldn't find a match. And then she remarked, there are literally millions of sequences in the database, and we're really shocked that it didn't match any of these species exactly in the database. And so, so she did that, and that was her results for the Josh Gates hair sample. Well, about that same time, while she's over here doing her thing and getting samples, my friend Dave Pilates, he was looking for a lab to test Bigfoot samples, to get something going, to forensically try to figure out what's going on here, get the DNA. And he was having a lot of trouble finding a lab that would test his samples. And basically, fate and circumstance brought David and Dr. Ketchum together, and they basically, based on Josh Gates' hair samples, she was definitely more open. And other samples she had been testing, to be honest, the way I understand it, Josh Gates got her mind open, and then she began testing other samples. And then when David approached her, she was more than willing to test his samples as well. And so Dave finally, had a scientist with a lab that was willing to test the samples, and he was, was grateful for that. So my involvement began in 2010 when I was corresponding with David Pilates, and that led eventually. I'd read his book, The Hooper Project, and I was looking for a little help, some people to bounce things off of, and things were starting to happen in what I was doing in my research that I was like, whoa, I need some help here. And so I started corresponding with David, and then and we kind of went back and forth, and I started working on, I think even before I was invited for NABS, I began to collect hair samples, but then in February of 2011, David invited me to join the team, and I accepted. So what we were trying to do, one of the things, the hair trap we came up with, and I've told this more than once, but we Dr. Ketchum had discovered that you have to have, in order for the hair samples to amplify, to yield any DNA of a Sasquatch, you have to have a skin tag. You know, like when you pull out the hair, it has the follicle and the skin tag with it. So you got to get that skin tag in order for you be able to extract DNA from that sample. That's mitochondrial DNA, which comes from the female. Comes from the female lineage. And because of this, and they didn't want to waste, they'd already wasted some money on just doing hair without the skin tag. So we were trying to figure out how in the world are we going to get hair, pull hair out. We needed to pull it out. It had to be pulled out by the root, so to speak. So came a little brainstorming, and we came up a way. Basically what we did is we took clear packing tape and wrapped it around the tree, sticky side out, and then we would wrap it around the tree and put, food or some sort of bait above it. And so when the Sasquatch would lean against it and reach, or we'd put apples and fruit in the tree, like a fork of a tree, 14, 15 foot up, and wrap the tape around, and then when the Sasquatch would reach up with his hand to get it, it'd pull the hairs off his arms. Or one of the very successful traps I had was during the cold period. I had what's called a feeding stations area where I would give them food and stuff, and it attract them. And I would take bacon grease, and I would smear it onto the tree, into the bark, about as high as I could reach, which would be about 8 foot tall, 8 foot up. And of course, you'd have to do this during cold weather, but but the grease, of course, would stick into the bark and on the bark and stay there. And then I would put down about the 4 or 5 foot level, I would put the hair, and when the Sasquatch would lean against the tree, and they would really go after it to lick the bacon grease and get the bacon grease off the tree. Of course, when they would lean against it, it would pull the hair out. And so, I would think it would hurt, but they didn't seem to care, and it was well worth the momentary discomfort to get the bacon grease. And one of the real successful ones was in the Smoky Mountains, and I had put some trail cameras out, and I had like one camera watching another camera. And what happened was I noticed the second camera was the infrared light was just at the edge of the throw. So what they would do, they have one camera looking to the right and the other camera looking straight at them, and they would set off the first camera, and they would get this tree. It was forked, and this tree right here is forked. And and what they would do is they would lean forward to look at this camera. You can see here, this guy, they would lean in, then enough of our light would reflect off their face, and pick them up. Now this is like a still capture from an actual nighttime, a 30-second video. And they would lean in and back out. It was kind of like, it wasn't like constant, like 3 or 4 frames you could make out the face, and then they would lean back and it would go dark again. And so what I did is, on this tree right here and on this tree, I wrapped tape around because they were leaning against that tree, and it was very successful. I got a big bunch of hair, but the funny thing was, I think I had it too low. And I think the hair caught them in a sensitive area below the waist, because when I went back and found it, and I've got a video where I've gone back in, I documented everything, but they had ripped the tape off the tree and thrown it. So I think that was kind of painful. And these are just different. These guys are probable hair donors for the DNA study. And so you got this ugly looking dude here. Oh, by the way, this little knob right here, that's 8 feet. So this creature here is right at his mouth is 8 feet. Oh, he's 9, 10 feet tall. And then you had some other characters that would lean in. There's the first one. You had these two guys that were one. This is 8 foot. Of course, you got one, and then you got the one with the Jimmy Durante nose over him. So this guy's 10 foot tall, 12. This guy's 8, 9. This guy's about 10. He's a different looking one. All you can see there are a lot of genetic differences between each one. These were on different. I think 2 of these were on the same night, and then the other one was on a different one. I have a really ghoulish looking one too that I posted before. Here's a good close-up of these two guys here, and you can that's infrared, but that's about as clear as you're going to get in infrared. Big old Jimmy Durante nose there, and and this is the hair sample I was talking about. This is the hair. This is Sasquatch hair, and this is I documented this before. I've got a whole video where I document the recovery and me packing this up and sending it off to the DNA study. Here's how we did the hair trap with the bacon grease. This is exactly how I did it. This tree I did it on, and I would smear the bacon grease on the bark and in the bark, and then put the packing tape. Here I put it in 3 locations, like chest, waist, and then leg hair, I hope. And then here's another big wad of Sasquatch hair that I found. This was like a tack that it looked like I found near a bed where that one had been laying, and it didn't really have any any follicles, but I sent it in anyway because there were some other tests that Dr. Ketchum could run. Hopefully she's run them, but this is what a big wad of Sasquatch hair looks like. And this is black and gray. Here's some more. These are the ones that we pulled out, and you can see here there's a skin tag. This is some more hair. I'm just showing you different. This was the the actual Sasquatch hair. This is perfect deal here, because you can see here that's the skin tag, and so it was pulled out by the root. Here's some more. You see the skin tags here. So these were again Sasquatch hair pulled out by the roots. In fact, this is off a trail. You can see the hair here. I found sometimes these I made natural hair traps by taking a sapling and breaking it, and it would kind of, and this one was natural, and I actually I just happened to be walking along the trail, noticed that in this sapling was this one hair, and it came back. It's a Sasquatch hair. It's about 6 to 8 inches long. Probably off his leg. This was down a well-worn trail the Sasquatch walked. And so I started making those. I'd find a sapling, 3 or 4 foot high, and I'd break it so it would splinter at the top, and right again, all along the trails where they were walking. And I have a few samples that way. Not as successful as the tape, but still pretty good. This was from my backyard. They were visiting me at my house, and so they were straddling I've got a fence that's 5 foot, and they were stepping over and straddling. I was finding footprints where they were coming over and that sort of thing. So I wrapped this tape around the top of the fence post, and this one right here is more of it's a brown and black. This is white hair. And both of those were in. One of these 2 samples was in the study. One of them, the hair expert said it was Sasquatch hair, and they tested it. I think it was in the study. Here are my samples that were in the study. It's just the location, the circumstances, just to show you. This was taken, excerpted from. These were the sample numbers given in the study, and some of these were from Loudoun County, East Tennessee, Smoky Mountains. These Tennessee hair trap ones were around my home. I don't think the white one is in there. I don't think I sent it in. well, I did send it in. It was just wasn't used. And so anyway, you can see here. So, you know, we talked about this last week, the common misconception that Dr. Ketchum worked alone, and that's not true. She, you know, she collaborated with other individuals and expertise in forensic DNA analysis, microbiology. How are we doing on time? 10 more minutes. Microbiology, molecular biology, biophysics, and biochemistry. That's just to name a few. Here are the co-authors. These are scientists that did work on the study. As I said last week and week before last, the co-authors do exist. They were aware of the study. They did work on the study. They did submit stuff. They wrote parts of the study in their areas of expertise. They signed a release, a copyright release that Dr. Ketchum has. They also got to review and sign off again on the final paper, the final draft of the paper for submission for peer review. So there's multiple times these people have documented, you know, have documentation that they were involved in the study, and they contributed, and so. This is the different doctors that were involved, what they are, you know, their specialties. And I can't pronounce a couple of these people, I just can't, you know, like Dr. Andreas Holzenberg. And you can see here he's from Texas A&M. He had published over 30 papers. He was professor, Department of Biology, professor of biochemistry and biophysics. Dr. Fan Zhang, PhD, bioinformatician in the academic and institutional resources and technologies, and he's from the University of North Texas Science Center. Dr. Patrick Watkins, I can't pronounce that, but he was director of the Shreveport Laboratory of the North Louisiana Crime Lab Systems, technical leader in the DNA section. He has been employed at the crime lab since 1977. Dr. Prachito of Wayne State University in Michigan, molecular biology, the background that also includes evolutionary biology, microbiology, and biochemistry. Dr. Douglas G. Toller, he was a clinical pathologist at Hughley Memorial Hospital at Fort Worth, Texas for 30 years. Miss Alice Watts of Integrated Forensic Laboratories, and you can see all her certifications: forensic biology, medical technology, phlebotomy, background in forensic DNA and quality assurance. She was alumni of the University of Texas at Arlington, where she taught Introduction to Forensic Science Laboratory, Forensic Biology, DNA, and Methods in Forensic Biology Laboratory. David W. Spence was the Trace Evidence Supervisor, for Southwestern Institute of Forensic Scientists, Criminal Investigations Laboratory at Dallas, Texas. And then here these other individuals were contributors there. And then here's what parts of the paper they wrote. I'm just going to go ahead and it's on the screen. If you want to pause it and look at it, you can. And this list is not all inclusive. And there, same thing with the labs. There was a misconception that Dr. Ketchum ran them all at her lab, but she did blind studies and sent them off to these are the different laboratories. I've gone over that before. And again, you can pause the video and look at these different labs, but you know, these are not fly-by-night redneck labs, you know, in Louisiana or Texas. You know, we're talking USC, University of Southern California. We're talking Texas Southwestern Medical Center, Texas A&M, Southwest Institute of Forensic Science. So let's see how I'm doing. 5 minutes. This is important. This is the screening that they went through for the hair, blood, and saliva and flesh samples. And I think this is very, very important. And I'm going to read this verbatim because, you know, this helps, because a lot of people in the coming weeks and months, you're going to hear it was contaminated. The hair was, you know, with common animals. You know, it was bear and dog and pig and horse and wolf and whatever else. The hairs weren't Sasquatch hairs. It was all contaminated. You're going to hear, I mean, you're just going to be deluged with that crap. And so I wanted to read that section about the screening of the hair, how they did it, how meticulous they were, and the fact that they already had a pristine Sasquatch hair sample. Told the account many times from the Hooper Reservation, and it was a woman watched the Sasquatch reaching in like an old, and it was an old metal storage building getting garbage. She watched him reach in. He cut his arm. When he was pulling his hand back out, he cut his arm and left a swath of, you know how it is. You cut your arm on a piece of metal. A big wad of hair and a little bit of flesh, hair, and blood. And a policeman came almost immediately. You know, was coming to to her aid because she'd seen the Sasquatch. And it didn't care, you know. She was sitting there around the corner of her house watching it. It just kind of looked at her and shrugged her off. And so when the policeman showed up, he could hear the siren coming, and Sasquatch got his garbage bags and took off. So she saw the Sasquatch, watched him leave the hair sample. The policeman arrived, and like, here's the hair, fresh blood and flesh. So he gets his crime kit out, and they forensically, collect the hair sample, and an evidence bag, etc. So they know they had the pristine sample of a Sasquatch hair, so they knew exactly what a Sasquatch hair looked like. So David Spence, who was their forensic guy, he would compare the hairs that we would send in, all the different hair submitters, and to make sure that they were actually Sasquatch hair. And so below is from the study, and it's hair analysis. Hair samples were sent to the Southwestern Institute of Forensic Scientists, Dallas, Texas, for analysis. Mr. David Spence was who did this. The samples were evaluated visually, stereoscopically, and by light microscope to determine human or animal origin. Hairs that were classified as potential novel hominid, that's Sasquatch, were also evaluated for DNA typing potential by examining the root material. Only hairs that were not human in appearance and could not be identified as any other species were utilized in this study. The hairs were examined for a variety of microscopic features such as medulla, pigmentation, cortical foci, and ovoid bodies, cuticle and root and tip characteristics. The hair cuticle patterns of the select hairs were cast by embedding the hair shafts in a thin layer of clear fingernail polish. The casts were examined microscopically. Photographs and photomicrographs were taken of select hairs using the Canon G3 digital camera and a camera adapter, and that's the model, MMX3S, for Martin microscopes. So again, I go over the hair samples needed to have the skin tag attached. The hair samples were processed and not contaminated. And so that, that's one of the big fallacies. And so they also jumped on chain of custody, and to be honest with you, that's a misnomer. I mean, they're using the PCR test, and chain of custody doesn't matter. They're looking at the DNA that's inside the sample. It doesn't matter if it floated out of the sky, was found on a fence post, was found in the woods. They're looking at the actual DNA, and like if they grab the hair, they know what the hair they're looking for. So the chain of custody is not this big bugaboo that they make it out to be. So real quick, the DNA study used forensic methods to wash and decontaminate the hair samples. The process is documented in detail in the DNA study. The fact that the samples were not contaminated is apparent in the results of the analysis. And I've gone over this before in the TV special Bigfoot Files, the Yeti episode. Dr. Sykes confirmed and validated that hairs can be washed and remove the contamination. So I just went over 30 minutes and it cut me off. So anyway, I'm gonna end it up here. I was wanting to. Here is that picture. I think I showed it last couple weeks ago, and here I can go back here. I got out of whack here. So yeah, Dr. Sykes confirmed here. The fact is, hair is very resilient. You can clean any suggestion of contamination from the surface of the hair without damaging the DNA that lies within. This is something we have known in the past couple of years. So basically, some of the detractors of the studies themselves admit that they can decontaminate the hairs and test them. So I think this is a good place, to end part 1. When we get back in the next part 2, and start part 2, we'll start with the isolation of the DNA and kind of some of the extraction things and some of the fun stuff. Let me go forward here, talk about some flesh samples, and there's also blood. And they got blood from one of them decided to chew up somebody's downspout, and go through that. And so with that, we'll go ahead and end part 1. I'm going to insert some other stuff. I've got a very interesting video I'm going to do on the entity in the portal. I did a really deep dive on analysis there, and it's really hard to get a still capture to blow up, and so what I did was I took still captures of the regular frame size, and then strung them together. In other words, they were in sequence. But I took them and I deblurred those, and I adjusted them really good with your contrast, and you deblur them, and you sharpness and all that good stuff, contrast and brightness, and got them looking nice, where as clear as I could get them. And then I strung them back together in a short video clip, and then I pulled that clip into my my mind's gone blank, into my CyberLink director, which is my production program that has a bunch of tools on it to help me clean it up and enhance it. And then from there I cropped it and blew it up bigger, and enhanced it, and again made it sharper. And I had a bunch of tools where I could adjust different things, to like to denoise it, and so it wouldn't be so blurry. And so I took, and then, and I also blew it up, and it didn't distort as bad blowing it up that way from the actual MP4, the video format. And then I was able to get, blow it up and get the 4 frames enlarged and enhanced without it just pixelating. And it's really interesting because you can see movement, some head movement and arm movement of this entity that is in that portal. So probably that's what I'm going to do next, and then part 2, and then I'll do part 2 of the DNA study. So with that, I've blabbed for about 35 minutes, and I'm going to cut it off here. Guys, thank you for watching, and have a good week. God bless, and be careful.